Hello everyone, my name is Lukman Ismaela. I'm a doctoral student at Laris Laboratory, University of Angers. Today I'll be presenting our work on self-supervision learning for functional brain network identification. Supervised machine learning has seen large success in recent years, especially in the computer vision-driven medical imaging domain. One of the well-known limitations is lack of sufficient annotated data set, and we can see this in three spheres. First, there exists a number of rare medical conditions where it's very difficult to acquire enough image. Secondly, international community maintain limited distribution of public data set and limited clinical expertise to annotate the data set. Some of the recent strategies to address this problem include few shot learning, which involves understanding the underlying pattern in data from few training samples, and synthetic data generation, which could include data augmentation, generative models, and so on, and also transfer learning, which is the reuse of learned representation from a pre-trained model on comparative data set. Interestingly, looking at the overview of transfer learning, where in this case, as illustrated in this case, we could acquire say for example the ResNet or VGG model, which are already pre-trained model from a lot of data sets, which in this case are already labeled, and this model quite perform excellently with the initial tasks. So this involves retraining and fine-tuning these data sets with, of course, our medical fMRI data set, and we expect to be able to recognize a functional brain network because the idea behind this is to be able to transfer good representation. Some of the a very recent approach of our work where we demonstrate the use of transfer learning from healthy subjects to unhealthy subjects. This has been a very interesting approach because we know that it is easy to enroll healthy subjects in non-invasive medical imaging modality, and this particular case was illustrated for functional brain network identification in resting state functional magnetic resonance imaging. And the brain tumor of unhealthy patients was also observed to have limited impact in resting state signals, which makes the features learned from healthy patients transferable into an unhealthy learning process. As some of the observed limitations, we recognize that the clinicians still have to annotate the healthy data for which there is no relevant in neurosurgical procedure or any medical or clinical benefit. And just to be exact, it can take about on average 10 minutes to annotate healthy volunteer data and about 15 minutes to annotate data of unhealthy patient. And also the process is currently not the routine because of the need for an expert reviewer in every case. And therefore, we ask a question that can we save the time of the clinician by using the healthy data without annotating them? An approach of this, one of the interesting approach as again illustrated here, is a possibility of self-supervision learning where we can use at the stage one, we can use unhealthy data. It could be really a lot of unhealthy data because in this case there is no prior need of annotating the large data set, and this is followed by a pretext task which actually earn us the pseudo labels, in this case angle rotation, and we train this via a convolutional neural network to be able to learn the representation in this. And of course this is done by passing it through a rotation classifier as demonstrated in this case. This is further in stage 2 trained with a linear classifier on the learned feature from the initial stage with very few label data. And Furthermore, in this third stage, we fine-tune this model for the complicated or the intended downstream task. In this case, it's object detection, or it could be classification or segmentation. What can you? So, learning good representation is demonstrated in several of these ways. And as an indication, we acquired data via the blood oxygen level dependent imaging system, which in fact is a way to understand the oxygen or energy consumption of our brain. And this actually shows the activated regions of the brain, otherwise referred to as the functional brain network or the biological network. An earlier study also has demonstrated the use of resting state case, where the patients do not need to perform any cognitive task to be able to understand the regions, the active regions of the brain. As a summary, we acquire 82 healthy control subjects and 55 unhealthy patients, and 55 features were extracted from independent components. 5 of these correspond to 7 functional brain networks, and each of these functional brain network map was labeled by an expert. As a visual understanding, here we have a sample slides from image samples of healthy data showing some of the networks and unhealthy data with an indication of lesion mask. Here it is expected that the lesion mask shows the region of the brain where the tumor is present, and it is observed that actual lesion is non-visible in healthy data. And in fact, the similarity in functional brain network image allows the transferability. Some of the very related recent work also demonstrate the use of self-supervision on fMRI data, which includes prediction of patient fatigue based on fMRI patterns and recognition tasks based on fMRI pattern visualization in healthy patients. And as an extension of our work, we propose to investigate the possibility of self-supervision and benefit and take advantage of the similarity between the healthy and unhealthy data set. And some of our experiments, which include the initial case experiments with healthy data, where we train a model with healthy data and test with healthy data, which gives us the first baseline result. Secondly, we also train a model with unhealthy data, and we test the model with unhealthy data, which gives us the second baseline result. And interestingly, we also approach the possibility of brute transfer learning, which involves healthy training and unhealthy testing. Now, the weight transfer involves using a pre-trained model to train and fine-tune on unhealthy data and testing on unhealthy data. And the very interesting self-supervision learning, which gives us the new baseline result. This one actually allow us to use healthy unlabeled data alongside with the unhealthy data, and we validated with tested with unhealthy data. As a general indication of the process of the experiments we just described based on self-supervision, it involves using of unlabeled healthy data following a described pretext task, where in this case we actually adopted the rotation, cropping, random translation, and so on. We trained our convolutional network, which gives the learned representation. And in the second phase, we actually retrain this model, the model with much fewer data sets. And of course, in this case, they are labeled unhealthy data set, and we fine-tune this data set for our functional brain network classification task. For our results for the experiment 1, which gives us the baseline value, we recorded 81.74 for SimCLR, where we trained healthy and tested unhealthy. The second one, where we trained unhealthy and tested unhealthy, we recorded about 73. And for the brute force, as well as not very encouraging was seen for the brute force and the weight transfer. And of course, lastly, the new second baseline result, where we are able to use unlabeled healthy data set. We recorded 76 results. It's important to indicate that the experiment on CNN gives the best result, but require the use of labeled data. And interestingly, similar performance range was recorded where unlabeled data was used in this case, and we also were able to save the 800 minutes of clinical expert time, which were normally used for annotating the healthy data set. So, as a conclusion, we demonstrated the use of self-supervision learning technique to avoid large data annotation. This technique was further illustrated using resting state functional magnetic resonance imaging data of healthy and unhealthy patients. We showed accuracy improvement of about 3% in training unhealthy data with unlabeled healthy subjects data for functional brain network identification task. This approach was observed to have significantly saved the time of the healthy data annotation, which is usually done by an expert. And finally, as a perspective, this method is applicable to any non-invasive medical imaging task for which healthy patients can be easily enrolled, as already explained, and there is similarity between healthy and unhealthy patient data. Thank you.